guys, so looking at our syllabus today, I have a quick little change, not a big deal, but looking at it today, we have chapter 4 lecture, and then I also have in there lab 17, our motility lab. With that lab, all we're going to do is look under the microscope and see different arrangements of flagella. So we're going to actually do that on Tuesday. Okay, so we'll do all that lab stuff on Tuesday. So today is simply lecture, and that is our day. Okay, so where it says check lab 17 results, it's just going to be the microscope work for lab 17. All right, and then as far as quizzes, hopefully you got busy on chapter 1 quiz, chapter 3 quiz. If not, again, you still have until, what is it, the 1st or no, the 2nd when we have our exam 1. So you have until then. Chapter 4 quiz, I already have it populated on Blackboard. So whenever you feel comfortable with chapter 4, you can go ahead and take that quiz as well. So again, all 3 are due June 2nd at the start of class. And then for next class, you can see that there are 4 labs due, so definitely, work ahead, answer what you can. Plenty of time in class to get it all done, but you want to work ahead just so it's efficient when you are in class. Questions for me? Okay, so let's start with chapter number 4. What I'd like to do is start off with a case study. So if you do have your textbooks, if you can open them up, that would be great. Page 81, where we see the start of the case study. Every once in a while, for a few chapters out of the book, I'll pull the case study out. We can just talk about it. It's kind of a nice segue into the chapter. So I'll have you go ahead and just read this first section out loud. Whole thing? Yep.
They're everywhere and they outnumber you 10 to 1. They are your microbiota. The thought of bacteria living on and in your body may make you squeamish, but these compatriots are vital to your health. Take the microbiota in your gut, for example. There are approximately 10 to the 14th power bacteria, archaea, and fungi living in your gut, accounting for about 1,100 different species, with about 10 to the 10th power cells per gram of feces. There are also about 1 billion viruses per gram of feces. The gut microbiota is a complex ecosystem comprised of numerous genera, including bacteroids, Clostridium, Streptococcus, Lactobacillus. I don't know that one.
Nice job reading those.
That's so good.
Yeah. By Phytobacterium.
By Phytobacterium and others. These organisms play an important role in your health, from helping to digest food, to producing vitamins and amino acids, to stimulating the immune system, to protecting you from harmful pathogens, to even having an influence on your behavior. Some recent studies have shown that the gut microbiota can have an influence in obesity. Scientists have found that the gut microbiota in obese individuals can extract more energy from the diet that is stored as fat. Other studies have shown that obese individuals have higher levels of lactobacillus species than non-obese populations. In studies with mice, scientists demonstrated that a change in diet from a low-fat, plant-based diet to a high-fat, high-sugar diet shifted the population of gut microbiota within a single day, favoring the microbiota that influence increased fat storage.
So you can see that there are, a ridiculous amount of bacteria that we do have within our gut, and that's just one location within our body. Looking at feces, a large, percentage of feces is actually living organisms, and these organisms present in our gut in order to prevent those bad guys, the harmful microorganisms from, initiating an infection. So they are definitely very, very important to have. And then things can happen when we eliminate those good guys from our gut, like in the case of C. diff, for example. And then, as we just heard, there are links to other types of diseases and influences from the types of microbiota that you do have in your gut. So let's keep reading, page 91. We're off page that one. Let's move it down the row. So it continues on page 91.
In a recent study, the fecal composition of normal, healthy adult males was compared with that of adult males with type 2 diabetes. Scientists used PCR to analyze the the 16S rRNA of gut microbiota and found that patients with type 2 diabetes also had elevated levels of bacteria from the phyla Bacteroides and Proteobacteria. Bacteria from these phyla are gram-negative, and this study suggests that the lipopolysaccharide outer membrane of these organisms may induce inflammation in the gut that could play a role in the development of type 2 diabetes. Yet another study suggests that multiple sclerosis, an autoimmune disease that attacks the myelin sheath of nerve cells, may be triggered by gut microbiota. Scientists found that mice raised without gut microbiota and then colonized with certain intestinal bacteria developed MS-like symptoms. Researchers still need to determine if a faulty immune system overreacting to gut bacteria is the cause of MS, or if a specific organism triggers the autoimmunity.
Right, those are just two examples where you can see links, to the types of, gut flora that are present in different disorders. But there's so many more out there, lots of research going on, and we'll just kind of move on down the road. That's where you can do that 3.
One important function of the gut microbiota is protection. The mere presence of billions of different bacteria prevents harmful bacteria from colonizing and causing infection. However, what if these helpful bacteria are removed? Often individuals taking antibiotics for long periods of time lose their protective gut microbiota and develop ulcerative colitis caused by Clostridium difficile and also known as C. diff. This infection causes severe diarrhea, nausea, vomiting, and abdominal pain, and is very difficult to treat because the organism has already been exposed to multiple antibiotics. The cure for some? Fecal transplants. Recently, gastroenterologists have suggested that a fecal donation from a healthy donor given via enema or through a nasogastric tube can reestablish a healthy gut microbiota and eliminate C. diff. Even though the practice has been documented since 1958, few doctors are willing to admit performing fecal transplants, even through this success rate of 90% or higher. The practice is gaining acceptance in the United States as C. diff infections rate increase. What is your poop worth? Apparently more than you thought.
Okay, so not a very pleasant thought, receiving a fecal transplant, especially through an NG tube, but looking at the validity of that, it's pretty amazing at that being one of the top treatments for C. diff, and the fact that C. diff patients have a really difficult time reestablishing their gut flora. Like, how do you get that bacteria back, So having that fecal transplant, knowing that, looking at the composition of feces, there's a ton of gut flora within the feces, and reestablishing that into another person, is a really kind of an ingenious way to go. So that's something you'll probably hear more and more about, as time goes on. So very interesting to look at, these bacteria and see what an influence they can have on our health. So kind of an interesting case study to start off with. And we'll begin chapter 4 with a little bit of a review. And, if you've taken the quiz for chapter 1, chapter 3, and read through these chapters and reviewed your notes, and even listening to lecture last week, this class is going to almost be like a foreign language with so many new terms that we're going to learn, and it's just going to keep building. There's going to be so many terms today in chapter 4 where you're like, oh my gosh, I've never heard that word before. I can't even pronounce that word, and I have to know what it means. It's just going to take a lot of time. If you're a fan of flashcards, that might be good for some of the definitions. You just gotta, figure out a way. I'll try and break down the words as much as possible to make it as easy as I can, but, repetition really is key with getting a lot of this down. The more you can look at it, the more you can read it. If you can explain it to somebody else, you have it. There's, nothing like trying to explain this information. If you can't, then you don't know it well enough. If you can explain all the different types of flagellum arrangements and show someone a picture or draw someone a picture, you know it. If you can't do that, you don't know it well enough. Okay, so those are some things you can try. Okay, so I don't want to scare anybody, but just definitely, it's just going to take a lot to, kind of get this down, especially in the beginning where there's so much being tossed at you and it's brand new type of content. Okay, so looking at the prokaryotes, we talked about how prokaryotes and eukaryotes differ. We also talked about bacteria and the archaea. So here we know that the prokaryotes, different ways, how they differ from the eukaryotes. Prokaryotes, pro means before, kary refers to nucleus, so we know our prokaryotes, they do not have that membrane-bound nucleus. They do have a nucleoid region. That's just where their chromosome is found, but it's not enclosed with any type of an envelope, any type of a membrane. Okay, so they do not have a set nucleus. We're eukaryotes. The EU, again, means true. Kary means nucleus, so they do have a true nucleus. Also, I'm just going to hop down here to this bottom one for just a moment. Prokaryotes, again, they do not have membrane-bound organelles. And again, by membrane-bound organelles, we're talking the endoplasmic reticulum, Golgi apparatus, mitochondria. Okay, so these elaborate organelles with those membranes. Prokaryotes, these bacteria, do not have those structures. Again, how do they undergo metabolism? How do they go, through all of these different types of reactions? They do a lot of this across their cell membrane and a lot within their cytoplasm. They just don't have those fancy organelles that eukaryotes possess. Okay, coming back up here, looking at the cell wall. First off, not all eukaryotes have a cell wall. Okay, we are considered eukaryotes. We do not have a cell wall. Plants are considered eukaryotes. They do have a cell wall. Okay, so not all eukaryotes have one. If you're looking at prokaryotes, depends if you're talking about bacteria or if you're talking about archaea in regards to the composition of their cell wall. There's a big new word up here. Bacterial cell walls are composed of this chemical. It is called peptidoglycan. That is what makes up the cell wall of bacteria. We oftentimes refer to it as the PG layer. P for peptido, G for glycan. Okay, but that is a chemical which is going to make up their cell wall, the bacterial cell wall. The A stands for archaea. And remember, the archaea, they're ancient, they're extreme, they've been around for a long time and live in these really harsh environments. So their cell wall, they have other chemicals that allow them to live in these bizarre, harsh, extreme environments. So they don't have the standard peptidoglycan cell wall. Okay, so just a little review of what we discussed last time. The rest of this chapter, we are going to explore prokaryotes. Essentially, we're really going to look at bacteria, I'd say for 90% of this lecture, and then right at the end, we'll just talk about the archaea briefly. But we're going to attempt to talk about bacteria like we are doing a dissection. So here we can see a nice flow chart, and we're going to start from these external structures with the bacterial cell, and then we have the barrier structures, and then we're going to talk about the internal structures. So we're going to attempt to dissect it and see what these bacterial cells have. So that's our plan for the day. Here's an awesome picture from your book. You zoom in so we can see this. It's kind of blurry up here on this screen, but bottom line, it shows a nice picture, a nice composite image of a bacterial cell, and it highlights a number of different structures. I'm going to expect you guys to know what all these structures are. This picture is fantastic because it gives you like a one-sentence summary definition of these structures. I think it's on page 82, maybe? 83. Okay, so this picture's on page 83. And the way they have it set up, right here it says in all bacteria. So these 4 structures are in every single bacteria. Okay, so you have your cell membrane, your chromosome, ribosomes, and cytoplasm. That's in every single bacteria. And then in some bacteria is everything else. So all these other structures are in some bacteria. And these 2 down here in yellow, these are also in some bacteria, but they're just not on the picture. Okay, I just don't show these on the picture, but all the rest, they show on that composite bacterial cell. Okay, so definitely know what all bacteria have, and then we can see what some may have. All right, so before we begin our dissection, what I like to do is just go over some terminology when it comes to shapes of bacteria, shapes and arrangement. So we're going to talk about morphology. So that's going to be the study of the shapes of bacteria. And we talked about this a little bit in the last lecture when we talked about how to name bacteria and how some of the names actually make sense based on what they look like. So we're just going to review that and add to it a little bit. So first off is the term coccus. Coccus, this term is singular. Cocci, C-O-C-C-I, that is plural. What coccus or cocci refers to is circular. So if you see the term coccus at the end, so if you hear micrococcus, you know that if you were to look at that bacteria in the microscope, you would see circles. Staphylococcus, streptococcus, all circular shaped bacteria. There are some circles. Okay. They're not always going to be perfectly circular. This one's a little bit pointy, but all in all, these are just different examples of cocci. There can be different arrangements. Okay, so up here, I have the term tetrads. What does tetra refer to? What number? 4. Okay, so if they appear in tetrads, that means they exist in groups of 4. Okay, so you would see actually 4 cells. There's existing in these packets of 4. There's another term down here, sarcina. Sarcina refers to grouping together in packets of 8 or 16 and sometimes 32. Okay, so in multiples of 8, essentially, for the sarcinae. So tetrads, groups of 4. Sarcina, you're going to see groups of 8 and multiples of 8. One more thing I want to show you is the term staph. Okay, the term staph refers to clusters. This refers to grape-like clusters. So you would actually see a bunch of cocci just in a big cluster. So the bacteria Staphylococcus aureus, it would exist in these clusters of circles. The term strep. For Streptococcus pyogenes, for example, Streptococcus pneumoniae, strep refers to chains. If it's Streptococcus, you would see chains like a pearl necklace, like so. Okay, so that's your strep. Staph is going to be in a cluster. Okay, so let's move on to rods. You can refer to them as rods, you can refer to them as bacilli, the term bacilli or bacillus. Bacillus is singular, bacilli is plural, but they are all rod-shaped. There is a genus, which is Bacillus, and that's actually the genus we worked with in lab on Tuesday. We did some inoculations using Bacillus megaterium. So you know if you were to look at it under the microscope, okay, it would be rod-shaped. And we'll see it under the microscope on Tuesday, because you guys are going to actually stain that bacteria. And you're also going to stain Staphylococcus. Okay, so you're also going to see circles in clusters. So rod, bacilli, looking at some images, they're all roughly a rod shape, as you can see. Some are a little bit more club-like, some are a little more like filamentous in appearance. But all in all, we're looking at basic rod-shaped. Here's that term strep again, so strepto, bacilli. So we know strepto refers to chains, bacilli means rods. Okay, so streptobacillus, you would have chains of rods. Streptococcus, you'd have chains of circles. So here we can see connected end to end, these different rods, kind of hanging out in these long chains. It's just sitting on some type of a filter paper. So those are just little pores in the filter paper. But the bacteria are the actual rods. Coccobacillus is a term hardly ever used. It's just when you have an organism which is sort of right in between a coccus and a bacilli. Rarely ever used though. In this class, it's going to be one or the other. Okay, now we're going to, let me move back one slide. We're going to talk about curved and spiral shaped bacteria. So curved and spiral. First one that we have, is going to be spirilla, or spirillum, singular. And for this one, so for spirillum, they are going to be thick, spiral-shaped bacteria. Not the greatest artist. Attempt to draw this guy. Okay, so pretty thick like that. So thick, spiral-shaped. And they always have flagella. And on this one, I drew flagella at both ends. Sometimes they only have the flagella at one end. So that's your key characteristic with spirilla, is that they're going to be thick, as you can see, and they have flagella. So that corkscrew type shape. Here you see them, and they all have this external flagella, as you can see. This one only has it at one end. These other two have it at both ends. Here we see a micrograph, so looking at a microscope, of this bacteria. The kind of the bluish color right here, bluish-green color, is the spiral-shaped bacteria, and then the brown would be the flagella. Spirochetes is next. Spirochetes are also spiral in appearance. They tend to be a little bit more like springy looking. They tend to be a little bit thinner. Okay, so there's one. It's a little thinner, a little more spring-like, and they have something unique. They have something which is known as periplasmic flagella. There is another term for periplasmic flagella. Axial fibrils or axial filaments, both terms for periplasmic flagella. On an exam, for example, I would use periplasmic flagella because it makes sense. Peri means to surround, plasmic, cytoplasm. So there's flagella that surrounds the cytoplasm, like it makes, that terminology makes sense. The weird thing about this flagella is it does not hang off of the cell. It's actually wrapped all the way around the entire organism. It would be like me taking a string and wrapping it around this bacteria and pulling it tight, or taking a string and wrapping it around my arm and pulling it tight. Nothing is dangling off of the cell. It's pretty easy to see how the spirillum would move because the flagella spin around and propel it forward. How does this one move if it just has a flagella wrapped really tight around it? Similar to a worm. Okay, it's gonna kind of like shimmy across, its environment. Okay, a little bit of twisting, flexing motion to move. Those are spirochetes. So you can see them look a little more bouncy, I suppose, more thin. You do not see external flagella. Here you can see them again on filter paper. Vibrio. Vibrio is a curved rod. Vibrio looks like a comma. Okay, so it's just going to look like, let's get rid of this guy. There it is. Just a rod that has a little curve to it. That's referred to as a vibrio. So thinking of comma shaped, that might be good. Flagella on that one. So here we can see some, they have a little curve to them. Some have flagella, some don't. This one doesn't, this one does. So they may or may not have flagella. This was vibrio. Here we can see a microscope image of vibrio. And this is actually a Vibrio cholerae, causative agent of cholera. And we can see a single flagellum extending from one end of these bacterial cells. A few more examples illustrating what we already talked about. So Streptococcus Coccus refers to circular shape. Strep, that means chains. So it's going to look like a bunch of circles connected end to end. Okay, so that would be streptococcus. Over here is a term we haven't mentioned yet. Diplo means two. Okay, so in your notes you should put down diplo means two cells. So if I ask you to draw me a diplobacillus, you should be like, okay, diplo means two, bacillus means rod-shaped. You would draw two rod-shaped cells end to end. If I asked you to draw a diplococcus, then you would draw two circular shaped cells connected end to end. Okay, diplo means two, coccus circular. E. coli likes to exist in this diplobacilli arrangement. E. coli is a rod-shaped bacteria, and they like to exist in pairs. Here's another term that's introduced to you in this chapter. This is not the greatest picture, but I can draw another one on the board. The term palisades. Palisades would be the type of an arrangement where you have rods. Okay, you'd see this with rods. And you would see them connected end to end, but they kind of snap back almost in a parallel arrangement. Not the greatest picture, but I can kind of draw a picture where I really illustrate this arrangement. So here you may have one rod-shaped cell. Here you may have another one. Here you may have another one. Here you may have another one. Okay, kind of connected end to end like so. This is palisades. Now, with this, they refer to it as a jackknife train arrangement because it has that jackknife train appearance. These are each train cars. And they also refer to it as the picket fence arrangement. It's a very common way to refer to palisades. And the reason they call it picket fence is because it looks like the top of a picket fence. If I just draw this, okay, so the bacteria up here just look like the top of a picket fence. So that's your characteristic palisade arrangement, where they're connected end to end, like so. Just a few more microscope examples. Here's an example of a spirochetes. Again, they do not have external flagella. They have that periplasmic flagella that you cannot see. Here is Borrelia burgdorferi, characteristic example of a spirochetes. Does anybody know what Borrelia burgdorferi causes? If I were to say the word tick.
Lyme disease.
Lyme disease. Okay, so this is the bacteria that the deer tick would carry, and then that's the bacteria that would cause that infection. Over here we can see a spirillum. So you can see it's much thicker, and you can definitely see the external flagella. And then the term pleomorphic, right here. Morph, again, refers to these shape changes. Pleo means many. So there's some bacteria, depending on their environment, they can change shape. Adjust their shape. This is most common in bacteria that do not have a cell wall. So what I'll write down up here. So I'll just give you a good example of a pleomorphic bacteria. We'll say mycoplasma. Mycoplasma, they have no cell wall, and they are pleomorphic. So they can change their shape because they don't have that peptidoglycan cell wall, so it's hard for them to maintain their shape. So their shape can change. Okay, that would be the mycoplasma. Yes?
Sorry, I just didn't catch it. Is the mycoplasma, is that a type of bacteria?
Yes, that would be a genus.
Okay.
Okay. Like Mycoplasma pneumoniae would be an example. A genus. Okay, before we go through the concept check, let me show you guys an animation that will wrap up this portion.
Karyotic microorganisms exist in a variety of different cell shapes. Some are spherical, but seldom are seen as a single cell. A spherical cell is called a coccus. Pairs of spherical cells are called diplococcus, the plural being diplococci. Chains of cells are streptococci, and grape-like clusters of cells are staphylococci. Some genera, such as Streptococcus and Staphylococcus, derive their name from the most common pattern of arrangement of the cells. Many prokaryotes are rod-shaped in appearance. A rod-shaped cell is called a bacillus. The genus Escherichia consists of rod-shaped cells. The genera Bacillus and Clostridium consist of spore-forming rods. Spore formation in some bacteria causes swelling of the mother cell. There are also many other genera of rod-shaped prokaryotes. Bacilli occasionally occur as chains of cells called streptobacilli. Cells of the Corynebacterium group have a characteristic club-shaped appearance. Some bacteria have a curved rod or vibrioid appearance. An example is the genus Vibrio. Mycoplasmas are a group of bacteria that lack a cell wall. The cells of this group are pleomorphic, meaning that the cells take on a variety of different shapes. There are two groups of spiral-shaped bacteria. Spirilla are motile by means of flagella, and spirochetes are motile by means of axial fibrils located beneath a sheath that covers the entire surface of the cell. Bacteria such as the nocardiae.
Okay, the other stuff we don't go into. So really nice review of the different cell shapes that are out there and a little bit of the arrangement of the cells. So definitely become familiar with those terms. You can expect me to have you pick them out or you guys will draw them for me, something along those lines with your exam. All right, so let's do a concept check. So 3 general shapes of bacteria. We have the circular shaped ones, which are referred to as coccus or cocci. What's the term for rod-shaped ones? Bacillus. And then we had 2 types of, what's the next basic shape?
Spiral.
Curved or spiral, yes. Okay, and within the curved or spiral, we had the vibrio, which are curved rods, the spirilla, and the spirochetes. 2 arrangements found among the general shapes of bacteria. What's one arrangement? Strep. Okay, so if you're looking at strep, would strep be, what would that look like? Okay, so strep would refer to chain. So streptobacillus, you would have chains of rods. Streptococcus, you would have chains of circles. What's one more arrangement? Staff. And staff would appear like clusters. Okay, so grape-like clusters, most commonly how we refer to it. What about diplo?
2.
That's 2. Tetrads are 4. And then we have the sarsenae,
16 through
16, and so on. Yep. And true or false? All bacteria have flagella.
False.
That is very, very false. Good. So let's talk about flagella. So with the flagella, in AP, for example, you learned about flagella, and there was one type of human cell that has flagella, and that would be the sperm cell. It's going to have its single flagellum, and that flagellum is going to kind of flutter around side to side and propel that cell forward. With flagella in bacteria, it's a little bit different how it's arranged, a little bit different with how it works. There may be multiple flagella, there may just be one, they may be found all over the cell, they may just be found at one end. So the arrangements can vary, and how they work is very different as well. So here's a general image looking at the 3 important parts of a flagella that we'll discuss. So I definitely want you guys to be familiar with these 3 parts. First part, and we're going to work from the barrier of the cell, so the cell membrane. We're going to work our way out. So the first part of the flagellum, and these are all made out of protein, first part is the basal body. The basal body is the anchor. This is what's going to embed the flagellum into the cell. Basal body is the anchor. pretty elaborate. We can see there's different rings and rods in here. You do not have to worry about the names of the rings or anything like that. Okay, just know that this basal body portion is what embeds the flagellum into the cell. We can see that it's spanning 3 different layers. It's spanning a cell membrane, a cell wall, and an outer membrane. With this example, this is gram-negative. If we were to talk about gram-positive bacteria, they don't have this outer membrane. And we'll get there in a little while. You guys will walk out of here today knowing the difference between gram-positive and gram-negative. You can look at this picture and tell me why it is gram-negative. But regardless, the basal body is embedded into the barrier structure of the cell. Next part, we have what's known as the hook. So the hook is next. And the hook, if you look at the shape of the hook, it's like an elbow, and it spins. This is the rotational part of the flagellum. Okay, this is going to spin around 360 degrees. Like a ceiling fan, like a boat propeller. Okay, it's going to spin all the way around. It just doesn't flutter side to side. It actually goes all the way around. And then the filament is the last part of the flagellum. And the filament is what you see. That's what hangs off of the cell. If you're looking at bacteria that has a flagella, the part that you're drawing, the part that you're looking at is the filament. Which, if this was drawn completely, the filament would probably go out the door into the hallway, looking at the length of it compared to everything else. Okay, so we're just going to keep on going. Okay, so basal body, hook, and filament. 3 protein components making up this flagellum. Next up, we'll talk about arrangement. And with these, it might be good practice just to draw them in your notes, just so you can have a little bit of practice. But we have 4 different arrangements for flagella. So trichus is going to refer to the flagellum, and then the first part of the word will tell you the arrangement of the flagellum. So first one that we have is referred to as monotrichus. Mono means one. There is one flagellum with monotrichus. So we're going to draw that. Here's the bacteria. There's one flagellum. Pretty straightforward, right? Monotrichus means one, so you just draw one single flagellum. All right, next up, we have LOFO. What in the world does LOFO refer to? Word out. So LOFOtrichus, LOFO refers to a bundle. A bundle or a tuft, or you can think of a ponytail. So now if I were to draw LOFOtrichus, I'll do that. That's LOFOtrichus. You know what else is LOFOtrichus? This is also LOFOtrichus. It might be at one end, it might be at both. Okay, so you're looking at LOFOtrichus. Next one we have is amphitrichus. The term ampha refers to both ends of the cell. So now, this is amphitrichus, because we have flagella at both ends. And you know what? So is that. This guy right here is actually LOFO and amphitrichus. It's both. LOFOtrichus because there's bundles, amphitrichus because there's flagella at both ends. This one is simply amphitrichus. You can't call it LOFO because there's no bundles. Could you call this one monotrichus?
No.
No, because I count 2. Mono means there's only one. Yeah, there's one at each end, but 1 plus 1 is 2, so that's not mono, okay, without a doubt. So those are your 3 types of polar. Polar refers to having flagella at one end or at both ends. Looking at the picture up here, we can see the 3 polar types. So this one, picture A, what type is this?
Mono.
This is definitely mono. What is this one? Picture C. amphitrichus, because at both ends, and then picture B, lophotrichus. Yep, because you see a bundle of flagella. All right, good. Next up, zoom this way. Okay, last one that we have is peritrichus. Peri means to surround. So peritrichus means you have flagella all around the entire cell. Peri, I mean it's everywhere. Okay, so peritrichus all around. And you might be thinking, well, if they spin 360 degrees, how does this bacteria actually go anywhere, right? Actually, peritrichus, this arrangement is the ultimate fastest for bacteria. The more flagella they have, the faster they're going to move. All of these flagella are going to work together. They're going to form one super thick bundle system and move that cell at incredible speed. So peritrichus by far is going to be the fastest. You can see that right there. That's peritrichus. You see it all over. And with flagella, it's really difficult to see flagella under the microscope. I'll show you guys on Tuesday. We'll take a look at what do I have. I have a peritrichus on a pre-made slide, and then I also have some amphitrichus on a pre-made slide we can take a look at. They're just kind of hard to see, but I'll explain how to find them. I think a couple of you might have looked at them when we did the pre-made slides on the first day of class. One of the best ways to check for motility is going to be to use this semi-solid media. So if we zoom in here, remember semi-solid media, just enough agar to allow them to move around. You don't want too much because that would stop their movement. They wouldn't be able to kind of penetrate through the agar, but you want just enough where they can move around and plus you can see their movement. Obviously liquid media, they're going to be able to move, but we would never be able to see them. Because as soon as you pick up liquid media, modal or not, they're going to move all over the place. So semi-solid is really good. This tube, you can see the student inoculated it straight down, drew the needle straight back up. Bacteria only grew where they put it, not modal. It only grew where they put it. Yeah, you can see some growth on the top because there was nothing to stop colonies from getting bigger. It doesn't mean it was modal. Where here, you can kind of see up at the top where they started their stab, but you can't really trace the stab because the bacteria were able to move all over this tube because they have flagella and they're modal. So this would be the modal bacteria and this would not be modal. It's a really good way to test for motility. Okay, so let me give you guys some definitions, just kind of clear some stuff up. Motility just refers to just the movement of bacteria, if they're able to move around. That's what we're looking at with motility. And you're looking at organisms being modal if they have flagella. Another term that I want to discuss is chemotaxis. Taxi means to move. Think of a taxi cab, think of a plane taxiing on the runway. Okay, taxi means to move. Chemo means chemicals. So chemotaxis is movement in response to chemicals. That's how bacteria actually move. They move in response to chemicals. They're going to move towards the nutrient or they're going to move away from some type of a repellent. Okay, towards something or away from something. It's all by chemical sense. They're sensing the nutrient, they're sensing the repellent. Again, they don't have eyes, they can't see it. Like if I wanted to walk out the door, I'm gonna walk right towards the door, I'm going to see the door. They don't see the nutrients, they're just kind of sensing the nutrients. Okay, so they're getting attracted to these nutrients or they're getting repelled from some type of, something harmful like a repellent. So with chemotaxis, we can talk about a little more specificity with the way the flagella work. Remember I mentioned that they're going to spin all the way around 360 degrees, right? Well, they can spin like this. Just going to zoom in on this a little bit. Okay, in this direction right here. This direction, is this clockwise?
No.
Or is this counterclockwise? This is counterclockwise. So I'll put down the word run. Okay, so a bacterial run is when there is 360 degree counterclockwise rotation. Okay, rotation counterclockwise. 360 degrees. That's a run. A run is when they move straight. They move in a straight line. Okay, so when the bacteria are moving in a straight line, that's referred to as a run. That happens when the flagella is spinning around 360 degrees counterclockwise. Over here, we see that the flagella is now moving in a different direction. Here it's moving clockwise. This is referred to as a tumble. Tumble would be 360 degrees clockwise. Tumble is when they stop and change direction. And they change their direction. So they're sensing these chemicals. If they're moving in the direction towards a nutrient and they think they're moving towards and all of a sudden they don't sense it as well, then they stop, change their direction, and they continue to move towards it because they're sensing everything. You might think, bless you, if this is the nutrient here, okay, this little star, this is the bacteria, you would think they would go straight towards it. Doesn't happen. They know it's over here, so they're going to do this. I'm going towards it, I'm going towards it. Wait, I'm going off course. Then they stop, they change direction. Wait, I'm going off course, and they stop, and then eventually they get there. Because again, everything is by sense. And that brings us to one more point. Positive chemotaxis. Positive chemotaxis is when the bacteria move towards a nutrient. They move towards something good. Here's the bacteria. Draw some bacteria right here. There's the nutrient. Same image I drew before. So this straight line movement, is this a run or is this a tumble?
Tumble. Run.
A run. Okay, so straight movement is a run. When they stop right here and they change direction, that's your tumble. This is a long run. Here's a tumble, and then here's a run. They eventually get there. So with positive chemotaxis, one thing we can jot down, fewer tumbles, longer. This board is crazy because I write something and then it fades away. The color fades away. Okay, longer, it's driving me crazy. Okay, longer runs.
Oh, that's weird.
Isn't it weird? All of a sudden it's like not as red anymore. Okay, so fewer tumbles and longer runs, opposed to this one, which is going to be negative. Negative chemotaxis. Negative chemotaxis, they move away from some type of a repellent. Now they're trying to get away from something. So here's the bacteria. And look, I'm going to draw him going the other way. He's going to try and get away from this repellent, which is the R. So when they do this, they're getting away. Okay, so again, run, tumble, over and over. Run, tumble, run, tumble, run, tumble, run, tumble, run, tumble, run. Right off the bat, overall, we can say that there are shorter runs and there are more tumbles. So basically, when the bacteria is trying to get away from something harmful, they have a much more difficult time. Okay, more tumbles, a lot more difficulty. When they're trying to get towards something good, they get there with a little more ease. I typically use the scary movie analogy. Kind of toss it in right here. So the person running away from the bad character, right? You know the person can run, but all of a sudden, it's like they forget to run, they keep tripping, they keep falling, right? They just can't do it when they're being chased. It's kind of like the bacteria. They're trying to get away from this harmful component, and there's so many tumbles. And they get up, and there's more tumbles, and so on. But going towards something good, it's a little bit easier. Okay, so positive chemotaxis towards something good, easier to get there, negative, moving away from something harmful. And again, know the difference between a run and a tumble, which direction the flagella are spinning, if it's counter or clockwise. Yes?
So are tumbles like is it like a quick thing? Like I'm thinking like, you know, like when you go and then you just like, you quick turn, or does it take them a while to like turn?
It's relatively quick. Yeah, it's very, very quick. Does not take long at all. They're pretty efficient with their motility. So here, we can see this is actually peritrichous arrangement. This is what it looks like in the moment that the flagella stops spinning before it goes in the other direction. Okay, we can kind of see it all kind of stretched out. But when it is actually moving, they all wrap around each other and they form this very thick structure. Not the greatest image because you don't see them spinning around the other direction over here, which is kind of bad. But I believe what they're trying to illustrate with this picture is with peritrichus, all the flagella work together as one unit, okay? Opposed to, 12 individual units, however many are up there. And then down here, just a reiteration of what we went through. Here you see motile bacteria moving around when there's nothing attracting it or repelling it. Here you can see the bacteria moving with a purpose, like they're obviously trying to go from here to here. Okay, they're moving towards something, they're moving away from something. Here, they're just hanging out in this space. There's nothing drawing them one way or another. This image right here is exactly what I drew here. It's illustrating the difference of positive chemotaxis on the top, so moving towards an attractant, negative chemotaxis on the bottom, moving away. This picture used to be in the textbook, but they took it out when they updated the textbook, so which is why we drew these on the board, so at least you have that illustration. And of course, you can access the Prezi. And then finally, looking at the spirochetes. Spirochetes, again, spiral-shaped bacteria, thin, a little bit more flexible. They do not have external flagella. They have that periplasmic flagella, also known as axial fibrils or axial filaments. And here you can see a picture of one. This noodle-looking structure is the bacteria, and then the white is the periplasmic flagella. Okay, so it's wrapped around really tight. You don't have your 360-degree rotation with that. It's a little bit different. Here you can see Borrelia burgdorferi. These little filaments right here, these are the flagella, periplasmic. Here's one that got displaced when they isolated it and tried to stain it, so one kind of fell off. But otherwise, you see them wrapped around really tight. These little white things are just arrows pointing to the flagella. So that picture is right out of your text. This motility animation is just going to reiterate the 3 parts.
Most motile bacteria move by means of flagella. A flagellum is a slender structure about 15 to 20 microns long and about 20 nanometers wide. A typical rod-shaped bacterial cell is 2 to 3 microns in length. The bacterial flagellum is composed of 3 parts: the basal body, which is embedded in the cell membrane and wall, a short curved segment called the hook, and the portion that extends out from the cell called the filament.
Okay, and then of course you can see the different rings in here, but here we can see a difference between gram-negative bacteria and then gram-positive bacteria over here with these layers. And we'll get into that after we take a break. But before we take a break, let's run through this concept check together. So 3 parts of the flagellum. We just heard it in the animation. So what would that anchor? What is the technical name for that anchor?
Basal body.
Basal body. What about the part that spins around?
Hook.
That's the hook, and the part that hangs off of the cell?
Filament.
Filament. Yep. Okay, difference between a run and a tumble. So does a run or a tumble incorporate 360-degree clockwise rotation?
Tumble.
That's your tumble. Clockwise is your tumble. That's when you stop and they change your direction. So the run is when they're moving, in that forward motion, and that would be 360 degrees counterclockwise. For flagellar arrangements, you should be able to name those and draw them. So what is the name of the flagella arrangement where you see flagella all the way around the entire cell? Peritrichous. What about when you see only one?
Monotrichous.
That's your monotrichous. And then when you see it at both ends of the cell? Amphitrichous by definition means both ends. And what if you see either at just one end or at both ends, but you see a bundle? That's Lophotrichous. Good. And then what type of bacteria is going to have the axial fibrils or the periplasmic flagella? Spirochetes. Fantastic. Okay, let's take a break, and then we'll continue on with the rest. You guys can take about 10 minutes or so.